2 lectures. We've only had 1 lab. Remember that all the enrollment stuff gets a lot less people in here already. What do you think?
Yeah.
Is it going that bad? We're rough so far? So we're going to deal with all the enrollment stuff this week in lab. So if you are enrolled in the you're enrolled in the Monday lab, and you want to keep your spot, you've got to show up today, and you've got to be there right in the beginning. Or I'm going to assume you're not in that class. I'll drop you and give your slot to somebody else. And the same holds for Wednesday. So if you're in the Wednesday lab, you want to hold on your spot, make sure you come on Wednesday, be there on time at the beginning for lab. So what I'll do in the very beginning is I'll take roll, we'll see who's there still, figure out how many spaces there are, how many people want to add, who those are, make sure I get your registration priority dates and times. We'll have to sort through all that, so it'll take a couple minutes. We'll figure out who gets to add, and we'll move on from there. Okay? So if you are trying to add the class, and you have not shown me your registration priority date and time yet, make sure you have that available for class. Right? Don't start finding it when I see you and ask for it. If you already have it there, load it up on your phone, or if you've got it printed out, whatever, make sure it's there and available. If you don't have it, then I'm going to have to put you at the bottom of the list, which is not good. Right? You want to make sure you get the slot that you deserve your registration date. Make sense? If you can take either section, try to add Wednesday. If that doesn't work out, try to add, or Monday, then add Wednesday if Monday didn't work out. Okay? So we're done with that for now. Okay, last time we were talking about cells, cell membranes, what they're used for. Anybody have questions about what we covered last time? Nobody has questions. Hopefully it is kind of all reviewed for most all of you. Let's go ahead and look at, I think this is where we left off. We're about to talk about what membranes or cell membranes are made of. Sound familiar? Okay. They are made of lipids, primarily made of lipids. They're also made of proteins, carbohydrates. So lots of different chemicals make up cell membranes. This is the first audio, I assume this is an audio recording device, that I've seen in a long time that's not a phone. So back in the dark ages, you click the button and there'd be physical things moving inside, and I'd have all those lined up. And then it's like there was this one semester, it was like just phones everywhere. And now we're going back in time a little bit. Whatever works, right? Okay, so lipids, proteins, carbohydrates. Let's go through these. The main type of molecule we find in our cell membranes are phospholipids. Phospholipids. So again, I'm sure this is sort of a review for a lot of you, so we'll go through this a little bit faster, but I want to make sure you guys have this fresh in your head. So phospholipids are really unique chemically, especially considering that they are lipids. Half the molecule acts like a typical lipid, the other half does not. And it's because of this that they make for such great cell membrane molecules. So one end of a phospholipid molecule is actually polar, has a polar head to it. So when we say polar, then it has at least a slight electrical charge to it. Right? Like a battery. Think of a AA battery. Can you guys all picture AA batteries? Right? And it's got it's like a long cylinder and it's got a little bump on one end. Right? On a fresh AA battery, it's got a certain voltage, right? Like 1.5 volts. The button end is the positive end. Right? The other end is the negative end. You guys have all had to pay attention to that for electronic devices. You have to put your battery in the right way or it doesn't work. That's because the battery has polarity to it. It's got a charge distribution across there. All right? So one end of this phospholipid has a charge. It's got a polar head to it. You could also consider it hydrophilic. Hydrophilic. Hydrophilic, which literally means water loving. You could care less about water, but it gets along with water molecules. Chemically, it can interact with water. So that's what we call it, hydrophilic. So when you think hydrophilic, think polar. We're going to use them interchangeably here. Okay? The other end of the phospholipid molecule would be two non-polar tails. Non-polar tails, so it doesn't have a charge, and it's hydrophobic. Which would mean water-fearing, or it doesn't get along or interact with water chemically. All right, so each end of the molecule acts very differently. Good. All right, so here's the chemical structure for a phospholipid molecule. I don't care that you memorize that. Got a little drawing of one over here. They're usually drawn as a circle with two little lines coming out. The circle is the polar head. The lines are the non-polar tails. All right, so one end it can interact with water. The other end tries to avoid water. So you've got a glass of water or a beaker of water. A handful of phospholipids. You throw it in the water. What happens to those phospholipids? How do they orient themselves?
The hydrophobic will go inside while the hydrophilic will be on the outside, because
Go into the glass.
you know, if you drop it in the beaker, once they're inside, they will arrange in a way where like the hydrophobic tails will be like lining inside like a circle, and the heads will be on the outside.
That's right. So you'd have a ball of phospholipids, right? And the idea here is the tails try to avoid water. Well, the best way to do that is if all the tails get together, going towards each other, they can exclude water. And this is all surrounded by the heads that interact with water. So now you've got this sphere of phospholipids, and those heads are interacting with the water molecules that are all around it, and the tails all pointing in towards each other, towards the center of that sphere, are excluding water. It's dry inside that sphere. Okay? And so they'll naturally orient themselves that way. You don't need enzymes to set it all up and keep them there. They're not bound together, they're just associated together because of their ways of interacting or not interacting with water. If you were to make this thing bigger, right, now what's going to happen? What's going to fill in the inside of this now bigger sphere? Right? It can't be a total vacuum or else it'd collapse on itself. So now what we can do when we make it bigger is line that with another layer of phospholipids. Okay, so here we've got, on the right is that ball. This is what you're thinking of, right? So we've got this ball of phospholipids, all the tails face in. There's no water inside here, it's dry. But on the outside, those heads can interact with water, no problem. Over here, this is what happens if we expand that ball. We make it bigger. We need something on the inside there. We're not going to fill that with water, because that water would then come in contact with the tails. Instead, we go ahead and put another line of phospholipids on the inside. And so, imagine that sort of stretched out over a bigger sphere. We've got heads facing out, interacting with water. The tails face in. The next layer of phospholipids has its tails facing up, and then we've got heads lining the inside. And now we can fill the rest of that space with water. So water can interact on this side, water can interact on that side, but it's not in the thickness of the wall. And this is really the basis for a cell membrane. It's a phospholipid bilayer. Questions about that? That's not new to you guys, right? All right, so here's a bigger drawing of a phospholipid bilayer. All these sort of blue marshmallow-looking things, those are proteins embedded in there. But you can see all these phospholipids. You've got water on the inside, water on the outside. All right, let's take a look at some of the proteins associated with cell membranes. Some provide structure. They can provide structure, so we'll go back to our drawing of a cell. Alright, so we got our phospholipid bilayer, and all these purple blobs are proteins. Some of these proteins are bound to other structures, right? Maybe inside the cell, so that helps structure the inside of the cell. Can help give that cell some shape. Some of these proteins can even bind to other cell membranes in other cells, so anchoring cells to each other to give more structure. Let's take a closer look at this sort of cell-to-cell interaction. Here is an example of, we've got a bunch of cells right up next to each other. Right? You can see that their cell membranes are coming in contact. Where they do touch, they've got a junction there. And we're going to call this a tight junction, this particular type of junction. So there are proteins in these cell membranes that stick to each other. And they keep these cells right next to each other. They're together tight. So close and tight together that chemicals are not able to sneak between the cells. Right? So the cells that line your gut are held together with tight junctions. You don't want just anything sneaking between cells. Think about your small intestine. Right? One of its main jobs is to absorb nutrients. Get all that good stuff that's in your gut and get it into your body. So you might think, well gosh, why make it hard for things to get in? Why not have big holes or spaces between cells so all those nutrients can flow right in? Well, what's the problem with that?
Can't regulate it.
You can't regulate it, right? So you can't control how much of those good things are coming in. And then what did you say? What about bacteria?
I have a lot of bacteria that's coming into your cells.
I probably have a lot of bacteria, or one in general. Is this a personal thing?
No, I was only doing the
Oh, okay. It does, but equally with everyone else. Right. So when you ingest food, there's all kinds of stuff in there. There are bacteria, and there's fungi, and there's worms, and who knows whatever else you stuck in your mouth, right? And you don't want those things to get into your body, to cross past this layer of cells. And so you've got these cells held together nice and tight. So the only way to get in, or from one side to the other, is you have to go across the cell membrane. So it's a form of regulation. We're regulating what can get into our bodies. All right, so that's tight junctions. Adjacent cells held together nice and tight with proteins, keeping chemicals from going between the cells. Since we're talking about cell junctions, here's another one, gap junctions. This also involves proteins in the cell membranes that are joining adjacent cells together. But in this case, they're not keeping chemicals from going between cells. Instead, what they do is provide a passageway from one cell to the neighboring cell. So there's actually a connection in the cytoplasm. And this is great for cells to communicate with their neighbors, right? They're also called communicating junctions. So if one cell gets a message, that message easily passes to the neighboring cell through this gap junction, another type of cell junction. And then I've got a third one here to show you. Desmosomes. Again, proteins in cell membranes holding adjacent cells together. This one also does not control what goes between the cells. But what this does is it keeps neighboring cells as neighbors. So it's a really strong, durable junction which keeps those cells from getting ripped apart from each other. So in tissues that get a lot of physical stress, we're going to have desmosomes to anchor those neighboring cells together. For instance, in your heart. Your heart goes through a lot of mechanical stress all the time. It keeps beating, these really strong, forceful contractions. And if the cardiac muscle cells were not held together by desmosomes, every time your heart contracted, it could rip those cells apart a little bit. You would rip yourself apart. Bad news. Right? But we hold those cells together nice and tight, so when it contracts, it all stays intact. Questions about those junctions? Yeah?
So, in order, it would be like the tight junctions are the strongest, and then the desmosome, and then gap junctions.
I wouldn't say the strongest, but they all have different functions. So tight junctions keep chemicals from sneaking between cells. Gap junctions allow neighboring cells to communicate with each other. And desmosomes anchor neighboring cells together so they don't separate. Yeah?
Can there be more than one type of junction?
In one
So-
between two adjacent
Yeah.
cells? Yeah, so let's
Like
go
say
back.
Our skin,
Like what?
our skin.
Depends on what part, where you look in the skin, of what kind of junctions you're going to have. Let's go back to the heart, because I use that as an example for the desmosomes, right? You also have gap junctions in it, right? So when one heart muscle cell gets the message, it's time to contract, that signal zips over to the neighboring cells through those gap junctions so that all of those cells in the area get the message at the same time. You don't want part of your heart to contract and the rest ignore it. And so one junction actually between adjacent cardiac muscle cells is really complex. It's going to have both desmosomes and it's going to have these gap junctions in it. Okay? They're called intercalated discs, so it's this big, super complex structure. It's got all this stuff in it. Okay, anything else? All right. So protein functions, we talked about structure. Some of these proteins and cell membranes are enzymes, right? They catalyze chemical reactions. Some of these proteins will act as receptors, which allows cells to communicate with each other. Right? Some chemical messenger is out there, out and about. It could bind to a protein on a cell membrane that acts as a receptor, letting that cell know, hey, that message was out there, that means do something. Let's see some examples here. Am I going too fast? No?
Can you give me an example or just is that all enzymes in general?
Well, no, not all enzymes are in cell membranes, but some of them are. Right? You can have enzymes associated with cell membranes.
They're all enzymes in
As far as I know, all enzymes have a protein component to them. Okay, anything else? All right, let's look at some receptor examples. Okay, so up top here, we've got two neighboring cells. One of these cells releases a chemical messenger, a blue triangle. We're going to call that a paracrine. So a paracrine is a signal molecule that acts on neighboring molecules, or not neighboring cells. This neighboring cell has a little protein in its cell membrane, a receptor, that can respond to blue triangles. So when that blue triangle binds to that receptor, this cell now knows what's going on, receives the message. So that acts across between neighboring cells. Autocrines, when a cell releases a chemical message and it actually binds to a receptor on its own cell membrane. Right? So it passes a message to itself, which seems a little bit weird because you think, well, I already knew the message. Well, this autocrine could actually also be a paracrine. So maybe this cell somehow got stimulated, releases this message to stimulate all the neighboring cells, and it also stimulates itself. So it's a way for them all to give the same response. What do we have down here? Neurotransmitter. That's not me, that's blurry, isn't it? No? Oh, it'd be nice to say this.
A little bit.
It's more blurry than the rest.
That one actually looks the worst to me. Is there a focus thing on here? So is everything else. Oh, well, that says neurotransmitters. So in this case, we've got a neuron that releases a chemical message that acts on neighboring cells. So in fact, it's very similar to how paracrines work. And what's different is the chemical message is coming from a neuron, or the nervous system. And then it binds to a receptor right there. And then hormones are similar, but they're going to work through the cardiovascular system. We've got some cell that releases a chemical message, in this case, green rectangles. And that's going to enter into the blood, the cardiovascular system. Gets pumped throughout the body. It eventually meets up with the correct receptor somewhere else. Then that cell that has that receptor receives the message, can respond to it. Questions about these? Yeah, question.
Could autocrines be relevant then for positive feedback loops? If it's giving a message to itself?
It depends on what the original stimulus is and what the response is to that. So it could be a negative feedback loop or it could be positive. Right? So if that binding to that receptor causes whatever the original stimulus is to be greater, then yeah, that's positive. But if it dampens it, then that would be negative. Anything else? All right. Receptors, what else? Transporters. So some proteins in cell membranes act as transporters, allowing chemicals to go across the cell membrane. So let's take a closer look at that. Remember that cell membranes are primarily made of phospholipid bilayers. Right? Chemicals that are polar or hydrophilic do not squish through cell membranes easily. They can't get along with those nonpolar tails. Water molecules themselves, they can't just squish right through. Right? So they're going to need some of these transport proteins. All right, so one form would be channels. So some proteins in cell membranes form channels. They're literally tubes that go all the way across the thickness of a cell membrane. And they allow for water and small ions to pass. So water is a polar molecule, can't go on its own. Ions, by definition, are charged. They're not going to go through the cell membrane on its own either. It needs some kind of passageway. Well, the channel could provide that passage. Okay. All right, let's look at a diagram. Here we've got a phospholipid bilayer, and embedded in there is a protein that forms a channel. So here the channel's open, you can see this passageway, and some chemical could pass right on through that. Right? And you can see as it goes through, it would be shielded from these nonpolar tails. Keep in mind that these channels, even though this looks like it would just let anything through, they're very specific. So sodium channels let only sodium through. Calcium channels only let calcium through, and so on. Right? So they can all be very specific. And a lot of these channels are gated, so they can actually close under certain situations. So here it's not going to let anything through. And then it can open again and let's go pass through it. So throughout the semester we're going to be talking about channels opening, closing, in response to different stimuli. Alright, so let's take a closer look at the gated part. What actually controls the gated part, or what controls or influences when the channel opens or closes? Some of these channels are chemically gated channels. A chemically gated channel is going to respond to the presence of a certain chemical. Right, so maybe it's free calcium ions. When that's available, that triggers channels to open or close, for instance. Some channels are voltage gated. What does voltage mean?
Charge.
Yeah, that's a charge gradient or uneven charge distribution, right? So the difference in charge across the cell membrane, we're going to take a closer look at that a little bit later today. Right? But the charge on the outside is not necessarily the same as the inside. If that charge difference changes, becomes greater or less, that could trigger a voltage-gated channel to either open or close. We're going to see that's really important in the way neurons work, for instance. Some channels are mechanically gated, just by physically being bumped that can cause channels to open or close. For instance, touch receptors in your skin. That's responding to a mechanical stimulus, being bumped. Okay, everybody have that? All right, so we talked about channels as transporters. There are also carrier proteins. And at first they seem very similar to channels, but there's a little bit of a difference. All right, these carrier proteins, they also pass ions and small molecules, just like we saw with channels. But the difference is, carrier proteins, they're never open all the way through. It's not like you could look to one side and actually look all the way into the cell. They're either closed on the inside or closed on the outside at any one instant. Let's look at the diagram. Chart, whatever this is called. All right, so here we have cell membrane in gray, and we've got a glucose carrier protein embedded in the cell membrane. All right, so these little green hexagons, those are representing glucose molecules. Notice with all these steps here that this protein is not open all the way through at any one instant. It's either closed on the inside or closed on the outside. So how is this going to work? Well, there's a little active site in here for glucose. So if glucose comes in and binds to it, that triggers this protein to change shape. At that point, it will close on the outside, open on the outside, and spit out the glucose. Okay? Carrier protein. So functionally it's doing the same thing as a channel, but there's some steps to it. And again, it's not open all the way through. Right? An analogy I always think of is the Panama Canal. Who's actually been to the Panama Canal? Two of you? I've never been, but I hear it's really cool. Did you guys sail through it or just look at it? You looked at it? You sailed through. How was that? Does it take a while? Huh?
It took forever.
Yeah. Worth it though?
Yes.
To do it once or did you do it twice?
Once is good.
Once is good. Okay, so now we know how worth it is. Okay. So the Panama Canal though, so that's going to, link across that union between, Central and South America, and it's not open all the way through. So it's not just a channel that's all the way through, right? If it is, then we have this incredibly drain one ocean or flood the other one, right? Those bodies of water on either side are at different heights, right? Their tidal cycles are out of schedule or out of sync and so forth. And so you can't just have the whole thing open and expect to rush all the way through. So instead, you go through in little steps, right? So maybe it's open on one side and closed on the other. So you go ahead and sail in here, seal it off back here, pump it up with water, open that up, and go to the next section, right? And that's the part that takes forever, waiting for the pump of water to come. And actually, I have been told, maybe you know this since you went through it, that there are no actual pumps. It's all driven by runoff.
It's all right off, yeah.
How crazy is that? You can lift this enormous ship without a pump. That's cool, right? It was cool, wasn't it?
It was cool once I realized what was happening.
Yeah. There you go, yeah. All right, so kind of like carrier proteins. All right, let's move on. All right, so lots of different functions for proteins and cell membranes. Okay? All right, I want to sort of go back to the transporter part though. I want to take a closer look at movement across membranes, cell membranes. We're going to look at all the different ways the chemicals can go across. Okay, so movement across membranes. All right, there are a few different mechanisms that we're going to cover here. The first one we're going to talk about is simple diffusion. Simple diffusion. And we're going to see that this is a passive process. You don't have to add energy to the system to get it to work. The system already has the energy it needs. We're going to look at facilitated diffusion, another type of diffusion. This is also passive. Anything with the word diffusion in it is going to be passive. We don't have to add energy to the system. And then we have active transport. And as the name suggests, this is an active process. We will need to add energy to this system for it to work. Okay. We're going to go through each of these three to see how they work. All right. Before we look at simple versus facilitated diffusion, let's just look at diffusion and make sure everyone's got a good sense of what that's all about. Everybody done with this slide? All right. So diffusion. What is diffusion?
It's the movement of water through a semi-permeable membrane. It normally goes by the grid, so like it has to balance. So like if there's like more water outside and less inside, we'll try to maintain balance, I guess.
Sure. So it could be water moving from where there's more water to where there's less water through some semi-permeable membrane. I know where you got all that from, and it's really to explain a very specific part of diffusion, which we'll hopefully get to a little bit later. So that's example diffusion, but let's think of diffusion even more broadly than that. It doesn't have to deal with just water. What's diffusion?
It's a movement of molecules from a higher concentrated area to a lower concentrated area.
That's right. So if you've got a high concentration of something, of some molecules for instance, those molecules would tend to spread out to a lower concentration area, or they would move down a gradient, right? So when we say down a gradient, sometimes that's confusing to people, so let's kind of work that out. A gradient is where you have a difference in something from one area to another. It could be a gradient in a color. So if we looked in here and we're going to have a gradient from black to white, right? So it's very dark over here, and then it's less and less black until it eventually gets to white. So we've got a gradient in the black here. Does that make sense? That's okay with that? And when we say down a gradient, well, there's no up or down here really. What do we really mean by down a gradient? The down part's actually an analogy. Let's imagine we've got a ramp or an incline. And there's a ball in the middle of the incline. Right? This incline is actually at a gradient. This is really a gradient of heights. One end of the ramp is really high, the other end of the ramp is really low. So that's a gradient. Everyone see how that's a gradient? This ball, if I let go of the ball, is it going to roll up the gradient or roll down the gradient? It's going to roll down the gradient, right? So when we talk about things moving down a, say, concentration gradient, it's not downhill or uphill, right? But it's going from high to low, and it's really referring to this analogy. Everyone feel good about what we mean by down and up gradients? Okay. All right, so diffusion is when chemicals can move from a high concentration to a low concentration, go down a gradient. What's causing them to move from one area to the other? What's that?
They're bouncing off each other?
They're bouncing off each other, right? So we could all start bouncing. Are we going to spread out and move out? Especially those who are like, why are we doing this? This feels weird, right? Get out of here. Right? So all of these molecules, they're moving. Right? None of them are completely stationary, right? The molecules that make up our atmosphere, they're bouncing around. Right? Let's imagine that we have some odoriferous chemical, right, that's released in high concentration, maybe it's like a perfume bottle, or somebody farts, or whatever you want to think about, right? You guys want to think about the fart? The kids be like, yeah, fart. Let's talk about farts. Okay, so you've got this high concentration of some smelly molecules, right? And it's all concentrated, let's say, right in the middle of the room. Are they going to stay in a ball? No. Right? Those molecules are bouncing around. They're going to keep going in a straight line until it hits something. And then it's going to bounce off and go in a different direction. So over time, they're going to spread out. That's diffusion, right there. They're going from this high concentration where they were released to low concentration, over to your nose. Right? As it spreads out. The molecules that make up this table are actually vibrating also. They're not stationary. If they were stationary, what would be true about this table? Wouldn't exist.
It'd be at absolute zero.
Could exist, but it'd be at absolute zero. What does that mean?
0 Kelvin, negative 273 point. Yeah, Celsius.
One of those guys, right? All right, so what does it mean to be at 0 Kelvin or absolute zero?
No,
So,
nothing's vibrating basically. There's no energy in
yeah, there's no molecular motion, right? So the molecules are all stationary, right? Which would be a real problem. So I'd instantly get frostbite if I was touching it. We couldn't do it anyways. Yeah.
I was also gonna ask, does it also mean it couldn't potentially be changed into something else? Like you couldn't cut it and like make it into something else or change the chemical composition by heating it or?
Well, if you heated it, then it no longer would be at 0 Kelvin because heat itself is molecular motion. So to heat it would be to add molecular motion to it and would raise its temperature. If this was at 0 Kelvin and we cut it, we would also heat it up because just touching it with our knife that's not at 0 Kelvin would transfer some heat to it. Right? So we can't actually get something at 0 Kelvin because every time you go to measure it or mess with it, it heats it up to some extent. Maybe not by much, but it does, right? So it's sort of it's not a theoretical temperature, but functionally it almost is. Right? All right, so everything we're ever going to interact with, most likely, is has some heat to it, has molecular motion, and if it's a gas or a liquid, these particles can move around, and they'll tend to spread out. Right, go from high concentration to low concentration. So that's diffusion. Diffusion actually can work with other gradients as well. It's not just concentration gradients.